Hello students, welcome to NEET World. This is Narayani. I am your botany educator for both senior and junior inter. Okay, so we are discussed for the senior inter people respiration in plants. We start the chapter of respiration in plants. In that, two questions are very important for the IPE. In the corner of IPE, those two questions, what are they? Glycolysis and Krebs cycle. Most of the in previous paper years, these two questions are quietly repeating. So we have to read both questions, then it is may have 90% chance you may get this out of these two questions, one question for sure in your IPE paper, right. So we discussed the first question, we completed that glycolysis, and if you have any doubts please let me know. And today we will discuss about the Krebs cycle, right. So before I am going to start the session, I would like to announce very important news from our side, NEET World. We are started the long-term course from the July 1st onwards, which means yesterday onwards we started the, long-term course for 11th, 12th and also repeaters and also droppers. So for 11th and 12th, who are currently studying the 11th and 12th, so by taking the parallel coaching, you can succeed in your upcoming competitive examination of NEET and also EAP set examinations. By enrolling with one course, you will get the 5 benefits of the 5 entrance test, you can get the enroll for that courses. So you will enroll one course, you will get 5 entrance test exam courses free. So it is very limited offer you guys. For 11th, 12th, you can join parallelly by, for your IP along take the coaching from us. It is the best opportunity, because we are offering just for rupees 3,333 per month. It's so affordable, it's like just tuition fee. And what are the benefits you get by joining with us? So 400 plus live classes you will get, and recorded classes also available. If once you not get the session is you not get that point, you can read, you can listen multiple times. So that option is there, because we will give online access also, right. So these courses offer both offline as well as online. For offline people, you can happily come to our institute present in Narayani, Hyderabad. Those who want to join through online, please go through in our app name called MedWorld. There you can get access of all live classes there. And along with that, we are providing the DPPs, daily practice test, and also the periodic assessments. Quietly, it is best choice to choose our NEET World, and the well crafted study material is provided by us, and then one-on-one mentorship will be provided to you. So thereby what you will have, so if you have any doubt, you can post your doubt on the chat option in the online. If you are coming to here, the lectures are directly available to you, so you can get any question sorted out, so in a very good explanation. And also all classes are digital classrooms, and if you want to join offline with us, we are offering hostel for girls and also boys too. So it is a great opportunity to you guys, because it just rupees we are providing 3,333 per month. So this is affordable price, and it is for both offline and also online. So if you want to come to online, come to our NEET World Institute present in Narayani, Hyderabad, near metro station. Those who want to join through online, by calling this number, we will give you access in our app name called MedWorld. So whatever you have queries, please let us know. So thereby we will clear out all your queries. So this is one of the important news from our side. Don't miss to join in our, in our courses. It is quite benefit to you. If you really passionate to become a doctor and you want to see yourself in a top engineering college, this is the best platform for you, right. So here this is our Telegram group channel. By scanning this QR code, you can access our Telegram group channel. So what are the benefits you get in the Telegram group? So the daily quiz of biology, chemistry, physics and mathematics you will get. So by for the competitive examination, you have to write multiple mocks. How many mocks you will write, you will get the gem of the person. So you will be in the gem of that examination. So we will provide daily quizzes, and if you have any doubt in that, if you are solving that question, if you have any doubt, you can write your in the Telegram group, you can keep a message that you have the doubt in the particular question. So we will give you sorted out, because all faculty will be there in the Telegram group channel. And the live class schedules. So we are purely informing the when was the live, and then you can join on time with us. And what are the free resources we will drop there. and also one-on-one doubt solving session. As I said, every doubts of yours will be clearing out by joining this Telegram group, because you will post the message, then we will see us and then we will clear out all your doubts. Right? If you have any queries of our institute, of the courses, so please let us know by this number. Then we will give you the all information about us. Okay? Then, okay, let's start the session. So, out of 2 questions we complete the glycolysis, right? So, let's see the Krebs cycle. Before we are going to the Krebs cycle, so what is the pyruvate fate? At the end of the glycolysis, how many molecules of pyruvate is produced? 2 molecules of pyruvate is produced. 2 pyruvates are produced, right? Is a pyruvate is a 3 carbon compound or 2 carbon compound? It's a 3 carbon compound, right? So it is a 2 carbon compound. So if they asked what is the where was the glycolysis occurs, students? It is occurs in the cytoplasm. Okay, it is occurs in the cytoplasm of the cell. And then here see the fate of the pyruvic acid. As we are seeing that for the anaerobic organisms, the only process occurs is the glycolysis, right? For the aerobic organisms, it's not like that. The pyruvate undergoes the aerobic respiration, which means it is entered into the Krebs cycle, which in the mitochondria. Why it is entering only the mitochondria? There is different cell organelles, right? Then why it is not entering those organelles? Why it is entering only the mitochondria? If you have a question like that, right? See, just scheme this, the fate of the pyruvate, then you will get the, you can recall. In anaerobic respiration, when oxygen is not available, pyruvate will be converted into ethyl alcohol. In this is the aerobic organisms. Anaerobic organisms. In aerobic respiration, what will happen? Where oxygen is available. So, like us humans, animals, the pyruvate will be plants, right? So, pyruvate will be completely oxidized into carbon dioxide and water in the Krebs cycle. Where is the Krebs cycle happens? The locations are very important on the corner of the both competitive side and also for the IPE₂, right? So completely oxidized. The pyruvate is completely oxidized. Which place? In the Krebs cycle, it is completely oxidizing. In the organelle name called mitochondria, right? So we know that glycolysis is happening in the cytoplasm. So let's see the Krebs cycle now, which happens at the mitochondria. If you have a doubt that why it is only happening in the mitochondria, why not other organelles are not involving in this process, right? See this. So we'll discuss in very clear way. Okay, 1 minute. So see this diagram. So see this diagram, then you'll get good understanding point of view. Okay, 1 minute. Why it is only happening in the mitochondria? Because the enzyme which is the pyruvate can convert it into the useful form. The enzyme which is present in the mitochondria, which is present in the mitochondria. See, here is the glycolysis, the process. At the end of the glycolysis, the product is formed is pyruvic acid, nothing but pyruvate. At the end of the glycolysis, what is formed? So at the end of the pyruvate, 2 pyruvates are formed, right? 2 pyruvates are formed. It is completely oxidized and forming the carbon dioxide in the mitochondria. Right? So if they ask the exact location, it is in the mitochondrial matrix. For one glucose molecule, how many ATPs will be produced at the end of the oxidation of this glucose? 38 molecules of ATP is produced, right? What is meant by cellular respiration? The cell which is producing the energy is nothing but cellular respiration. To get the 38 molecules of ATP, to get one glucose to produce energy, it requires the glycolysis process and Krebs cycle. Then it will enter into the electron transport chain. These are the 4 cellular respiration cycles which are involving, which means glycolysis is involving, Krebs cycle is involving, and there will be link reaction, oxidative decarboxylation is happening. We will see this now in this class, and then electron transport chain. So first oxidative decarboxylation, then Krebs cycle, then Krebs cycle, then Krebs cycle, then electron transport chain. We studied out of this, cellular respiration, we completed the glycolysis. Then we have to complete the link reaction. Now we have to know about the link reaction, that is oxidative decarboxylation. Okay, then we will learn about the Krebs cycle. Then we will see the electron transport chain. Okay, glycolysis is happening in the cytoplasm. So at the end of the glycolysis, we are forming the pyruvic acid, right? How many pyruvic acid? 2 pyruvates are forming. So these pyruvates are entering into the mitochondria. That is why, see how the beautiful picture it is explaining. Here the 2 pyruvates are entering into the Krebs cycle, which is in the mitochondrial matrix. Then it is happening the electron transport chain, right? So we have to know about each and everything clear way. So now what is happening for the pyruvic acid? Let's see this. Pyruvic acid is a 3 carbon compound. How many carbon compound pyruvic acid is? 3 carbon compound. How many 3 carbon compounds are produced by the at the end of the glycolysis? 2, right? These are the 2. See, this is the cytoplasm. This is the cytoplasm region. Here at the end of the glycolysis, what formed? Pyruvate is formed, right? It is a 3 carbon compound. How many 3 carbon compounds are produced? 2 3 carbon compounds are produced at the end of the glycolysis, that is pyruvates, right? 2 pyruvates are formed. So it is why it is entering only the mitochondrial matrix, because the enzyme name pyruvate dehydrogenase enzyme. Pyruvate dehydrogenase enzyme, which is present in the mitochondria matrix. That is why the pyruvate is after the production of glycolysis, that the end product of the pyruvate is entering into the mitochondria matrix. Then with the help of this enzyme, the pyruvate is forming into the acetyl-CoA. So let's first see this, how it is happening and all that. Then we will learn about the Krebs cycle. How the Krebs cycle is happening. It is one of the very important question for the IPE. You should not take any choice for this. Okay, 1 minute. Okay, 1 minute. Okay, see this. This, the pyruvate is entering into the mitochondrial matrix, right? And that process, that action we call it as oxidative decarboxylation. Oxidative decarboxylation. This oxidative decarboxylation, which means pyruvate is converted into the acetyl-CoA. Oxidative decarboxylation. Oxidative decarboxylation. So oxidation is happening, right? And decarboxylation is also happening. What is meant by decarboxylation? Elimination of carbon dioxide is the decarboxylation, which means pyruvate is 3 carbon compound, right? It is a 3 carbon compound. By, when it is converting to the 2-acetyl-CoA, 2-acetyl coenzyme A. Okay, it is 2 carbon compound. These 2, wait, we will calculate for 1 first. So 1 pyruvate molecule is converting to the acetyl-CoA, it becoming the 2 carbon compound. How many carbon compound? 2 carbon compound. It is a 2 carbon compound, which means 1 carbon is eliminated now. So for 1 pyruvate is a 3 carbon compound, right? So for 1 pyruvate is converting into the acetyl-CoA, it becomes the 2 carbon compound, which means elimination of carbon dioxide is occur. That is why we calling as the oxidative decarboxylation. It is happening oxidative also, na, which means reducing equivalent is producing. Reducing equivalent, what is reducing equivalent? NAD plus is producing. So NAD plus is producing into the NADH, right? That is by oxidative decarboxylation. The elimination of carbon dioxide and the production of reducing equivalent NADH, right? For the one pyruvate molecule, it produces the one acetyl-CoA, right? It is a 3 carbon compound, it is a 2 carbon compound. One carbon dioxide is releasing out, which means decarboxylation is happening. That is why the name we call this reaction as a oxidative decarboxylation, which where it occurs in the mitochondrial matrix. In the mitochondrial matrix. Got it? So it is the coenzyme A, right? So here it is also happening the condensation process. Here the coenzyme A is adding to this process. Coenzyme A is adding to this process. That is why the pyruvate become the 2 carbon compound called acetyl coenzyme A. Here it is the coenzyme A is adding to this, and here is the elimination. Here it is what is happening, children? It is the elimination process is happening. This is the elimination, and it is the production of NADH, reducing equivalent, and here is the entering of coenzyme A. This process, condensation process, is happening, right? So then what is the enzyme? It is, as the in glycolysis also I said to you, whenever the NADH production is happening, whenever the NADH or FADH are reducing equivalents, whenever these reducing equivalents are forming, then there is a enzyme name called what will comes to that place, dehydrogenase enzyme. As it is acting on the pyruvate, this enzyme is called pyruvate dehydrogenase. Pyruvate dehydrogenase enzyme. Got it? So whenever the dehydrogenase enzyme came, there is a production of reducing equivalents, either the FADH₂ or NADH. Got it? So why we call this oxidative decarboxylation? Because the elimination of carbon dioxide, right? So so this is what happening in the cytosol region. So in the mitochondrial matrix, in the cytosol, the pyruvate is forming. The end of the glycolysis, pyruvate. Okay. So 1 minute. Okay. At the end of the pyruvate is forming in the glycolysis, in the cytoplasm of the end product of the glycolysis is pyruvate. It occurs in the place of cytoplasm. The link reaction is also called as oxidative decarboxylation reaction, occurs in the mitochondrial matrix. So whenever it is converting the mitochondrial matrix, for one pyruvate molecule is getting back to the forming of one pyruvate molecule converting into the acetyl-CoA. In the formation of acetyl-CoA, what are adding and what are eliminating? As it is a decarboxylation, as it was the process the decarboxylation, elimination of carbon dioxide, and it is the oxidative too, right? So here the NADH is producing, NAD plus is reducing to NADH plus H plus, right? And it is a condensation process is also happening. That is why coenzyme A is adding to it. That is why as a reducing equivalent, whenever the reducing equivalent is forming, there is a enzyme name called dehydrogenase comes to the picture, right? So as it is acting on the pyruvate, this enzyme we call it as pyruvate dehydrogenase. Why it is happening in only mitochondria? This pyruvate dehydrogenase enzyme is specially present in the mitochondrial matrix. Got it? If you not understand this reaction, you cannot understand the Krebs cycle. That is why link reaction is very important. So there are pyruvate is a 3 carbon compound. Acetyl-CoA is a 2 carbon compound because 1 carbon dioxide is reduced. Like that only, how many pyruvates are produced in the end of the glycolysis? 2 pyruvates are produced, right? Which means 2 acetyl-CoA are produced. Okay. So these 2 acetyl-CoA are also 2 carbon compound only, right? For one pyruvate, like that one carbon is how it is getting out, for another pyruvate is also same, the reaction is happening same. Got it? So then this acetyl-CoA get into the Krebs cycle. We'll see that. So Krebs cycle is also happens in the mitochondrial matrix only. So let's see the how it is happening and all that. So see, so here I am giving the very best easy way to remember the Krebs cycle. It is quite easy. Don't at all think that these are their typical names are there. They are so easy way if you understand means. So the pyruvate of the link reaction, what got formed? So pyruvate is turned into 2-acetyl-CoA, right? 2-acetyl-CoA. This process all happen in the mitochondrial matrix. Okay. So this acetyl coenzyme A. So it is going to the, so someone is have to accept it now. That is oxaloacetic acid. Oxaloacetic acid. Wait. Oxaloacetic acid. So I will give you the first, I will explain you how would you have to remember. Oxaloacetic acid. Always remember that, see, first OCCI. O, first write O, then C, then again C, then I. Remember O, first line you have to remember OCCI. Then you have to turn down. Then here it is a O and alpha. O and alpha. Here it is having the O and alpha. Okay. Then you have to write S square. Why I am saying this? I will say to you. So then S square. Then you have to again get back up. So here is the FM. F and M. So Krebs cycle is a cyclic process, which means the point where it starts, it is again going back to that place only. So first you have to remember OCCI. Write that in an examination also like that, and remember also like this. OCCI. Then O alpha. Then S square. Then FM. F and M. Then again getting back to the same place of oxaloacetic acid, it is formed. So see here, you have to remember like this only. Then it is very, very, very easy for you. So this oxaloacetic acid converted into the citric acid. Okay, citrate. 1 minute. Citrate. So then it is formed the cis aconitase. Cis aconitase. Then it is forms the isocitric acid. Isocitric acid. Then it is the O is oxalosuccinic acid. Oxalosuccinic acid. Then it is forming the alpha ketoglutaric acid. Then it is forming the alpha ketoglutaric acid. Then this S square is there, no? Succinyl CoA. Succinyl CoA. Coenzyme A. CoA means coenzyme A. When here it is the coenzyme A is coming, there is a process of condensation process happening. Remember that. So here is the S square, right? Succinic acid. Succinic acid. Then it is getting forming the fumaric acid. Then it forms the malic acid. So remember, this OCCI along with O alpha and S square, they went to listen the FM radio. Remember like that. OCCI along with the O and alpha with S square, they went to listen the FM radio. Remember like this. So see here. So which means here I drawn the 4 corners, right? So in these 4 corners there is a release of reducing equivalents. In the oxaloacetic acid, it is the releasing of the reducing equivalents, and this isocitric also releasing the reducing equivalents. This alpha ketoglutric also releasing the reducing equivalents, and this here is also producing the reducing equivalents. First see one on one first. So 1 minute, first I will write like this. Okay. Oxaloacetic acid. So first, what I said, in the four corners there is a production of reducing equivalents, right? So here first we will start with the isocitric acid, which means here is the production of reducing equivalents, right? So which means reducing equivalent means NADH is producing, NAD plus is reduced to NADH plus H plus, wait, NAD plus reduced to NADH plus H plus. Okay, got it? So here is a one reducing equivalent is produced, which means it is isocitric dehydrogenase enzyme is acting, because in the when the isocitric acid is converting into the oxalosuccinic acid, then only it is forming the reducing equivalent, right? That is the enzyme name. What we call the enzyme name is isocitric dehydrogenase enzyme. Dehydrogenase enzyme. Whenever the reducing equivalents are forming, there is a compulsory the enzyme name called dehydrogenase comes to the picture, right? So then next, this oxalosuccinic acid forming the alpha ketoglutric acid, right? Alpha ketoglutric acid. This alpha ketoglutric acid forming the succinyl coenzyme A, right? Here is the production of NAD. Here is the production of reducing equivalent. I said in the four corners there is a release of reducing equivalents, right? So this is the NAD plus is reducing the NADH₂. NADH plus H plus. Got it? Which means the enzyme acting is the alpha ketoglutric dehydrogenase enzyme. Dehydrogenase enzyme. Got it? Okay, whenever the reducing equivalents are forming, then it is a definitely came to the picture of dehydrogenase enzyme. Coming to here the fourth, here the third corner. So succinic acid is forming the reducing equivalent. Which reducing equivalent? FADH₂. In the first starting of the class, what I said, NADH and FADH are the reducing equivalents, right? Which means they are indirectly producing the energy, that's all. They are also producing the ATP only. So here is the succinic acid to fumaric acid, there is a production of FADH₂. FAD plus is reducing to the FADH₂. FADH₂. Got it? So here is the enzyme name is succinic dehydrogenase enzyme. How easy it is, right? Succinic dehydrogenase enzyme. Succinic dehydrogenase enzyme. Got it? So in the four corners the reducing equivalents are forming. So on the fourth corner is oxaloacetic acid. Here is the forming of NADH. NADH plus is reduced to forming of NADH plus H plus. Right, here is the enzyme is malic na. That is why malic dehydrogenase enzyme. Malic dehydrogenase enzyme. Malic dehydrogenase enzyme. Got it? So this is how the reducing equivalents are formed. So let's see the another having the So which enzymes? We know the four corners, which enzymes are produced, right? These are the reducing equivalents are produced at the four corners. Let's see. Oxalic acid is how many carbon compound? It is a 4 carbon compound. It is a 4 carbon compound. When this acetyl CoA combines with this oxaloacetic acid, 4 plus 2, 6, 6 carbon compound, which means it is combining with oxaloacetic and forming the 6 carbon compound, nothing but citrate, right? Nothing but citric acid, citric acid. So here it is the 6 carbon compound. So cis acnotase is also 6 carbon compound only. Isocitric acid also 6 carbon compound only, right? So which enzyme acting to forming the cis acnotase? This is acnotase enzyme, right? So here is also enzyme name is acnotase. Got it? Then see. Here, so along with their went to listen the FM radio na. Here the water molecule also want to come inside along with them, but citric acid not accepted it, so it is sent out the water molecule, again it is not give up, it is again added by the here water molecule is added in the when is the cis acnotic acid form the isocitric acid. Okay, so now, which enzyme is acting for the isocitric acid to oxalosuccinic acid? That is isocitric dehydrogenase enzyme. Then oxalosuccinate to alpha ketogluteric acid. Here it is the this is also 6 carbon compound and it is reduced to 5 carbon compound. Alpha ketogluteric acid is the 5 carbon compound. Got it? So here is the succinic acid. Here is it is a 6 carbon compound, right? It is a 5 carbon compound. What is happening? 1 carbon dioxide is eliminating, right? Which means decarboxylation is happening, right? So here is the enzyme name called, wait. So here it is the enzyme name called oxalosuccinic decarboxylase enzyme. Decarboxylation is happening na? That is why oxalosuccinic decarboxylase enzyme. Got it? So quite easy for you. Remember, I am writing like this. I will again draw in a clear way to get few people understand for you. And then this alpha ketogluteric acid, how many carbon compound? 5 carbon compound. Then it is turning it back into the 4 carbon compound. Succinyl coenzyme is 4 carbon compound. Then there is a release of carbon dioxide here, right? And it is having the coenzyme A right now, which means the condensation process is happening, which means coenzyme A is adding to this process. Got it? Then this succinyl CoA forming the succinic acid. Here it is the forming of GTP, which means substrate level phosphorylation is happening. Here it is the forming of substrate level of phosphorylation. Here is the GDP combines with inorganic phosphate means IP, then it forming the GTP. GTP is nothing but ATP only. In animals GTP is formed. Okay. In plants ATP will be formed. Okay. Then it is forming the fumaric acid. Succinic acid forming the fumaric acid in the presence of enzyme name called succinic dehydrogenase enzyme. Then it is getting back to converting the malic acid with the enzyme name called fumarase. Enzyme name called fumarase. Here is the one water molecule is adding. Then this malic acid forming the oxaloacetic acid. There is a completion of cycle, right? So where it is started? With oxaloacetic it is started. Got it? All of you? So first oxidative decarboxylation, happening in the mitochondrial matrix. In that pyruvate is converted into the acetyl coenzyme A. Then the acetyl coenzyme A is welcomed by the oxaloacetic acid. So this oxaloacetic acid get converted into the citric acid. So then citric acid is converted into the cis-aconitic acid, right? 1 minute. Cis-aconitic acid. Wait. Then this cis-aconic acid forms the isocitric acid. isocitric acid. This isocitric acid forms the oxalosuccinic acid. This oxalosuccinic acid again form the alpha ketogluteric acid. Then this alpha ketogluteric acid forming the succinyl coenzyme A, right? Then this succinyl coenzyme A forming the succinic acid. Then it is forming the fumaric acid. It is again getting back into malic acid. This malic acid turns into the oxaloacetic acid. What are the 6 carbon compounds here? Citric acid, cis-aconic acid, isocitric acid, oxalosuccinic acid. These are the 6 carbon compound. Only one 5 carbon compound is alpha ketogluteric acid. This alpha ketogluteric acid get converted into the 4 carbon compound, that is succinyl coenzyme A. Okay. So this succinyl coenzyme A, succinic acid, fumaric acid, malic acid, these are all 4 carbon compounds. Got it? So here how it is happening, this is called Krebs cycle. So why it is called Krebs cycle? So why it's not any other name? So it was on the memory of the scientist Krebs. He got the Nobel Prize also. On the memory of him, this cycle is Krebs cycle. Okay, this cycle is also called as tricarboxylic acid cycle. Why it is called tricarboxylic acid cycle? Which means TCA cycle. Why they called TCA cycle? Which means in citric acid cycle there is a tricarboxylic acids are there. That is why it is also called as tricarboxylic acid cycle. Got it? And also this cycle also called as citric acid cycle, because the first compound is formed by the combination of acetyl-CoA with along with the oxaloacetic acid. What is the first compound is forming? Citric acid, right? That is why it is also called as citric acid cycle. Got it? Why it is called Krebs cycle? Why it is called TCA cycle? Why it is called citric acid cycle? In the examinations, if they ask citric acid cycle, you don't think that I read the only Krebs cycle. Don't think like that. Krebs cycle or TCA cycle, citric acid cycle, 3 are same. 3 are producing the cycle. These 3 are same, which is oxidizing the carbon dioxide and water here. Got it? So first today you skim this all this, then tomorrow we'll discuss how many carbon compounds are formed. So how many ATPs are produced here, how many NADH are produced for one pyruvate, we will discuss in the tomorrow session. So before I am winding up the class, I would like to tell you one important news. So I would like to say one important thing. In the among all, one is the 5 carbon compound. What is that? That is alpha ketogluteric acid is the 5 carbon compound, right? Alpha ketogluteric acid is the only 5 carbon compound present in the Krebs cycle. Got it? So 4 carbon compounds are succinyl coenzyme A, succinyl coenzyme A and succinic acid, fumaric acid and malic acid, and one of the most first, when acetyl-CoA is combined with the oxaloacetic acid na, so that oxaloacetic acid is also oxaloacetic acid is also 4 carbon compound. These are the 4 carbon compounds are produced in the Krebs cycle. And what are the 6 carbon compounds? That is citric acid, isocitric acid, citric acid, okay, cis-aconic acid and and isocitric acid and oxalosuccinic acid. Oxalosuccinic acid. These are the 6 carbon compounds in the Krebs cycle. Only 5 carbon compound is alpha ketogluteric acid. Okay. So I said you to remember like this now. OCCI. Just write those in the first line. Then O and alpha. Then S square. They went to listen the FM radio. Then again that getting back into the own house. Right. Oxaloacetic acid. Got it? OCCI, O alpha, S square. They went to listen the FM. Along with the OCCI, along with the O alpha with S square, they went to listen the FM radio. Got it? So in the 4 corners there is a production of reducing equivalents, right? In the 4 corners there is a production of reducing equivalents. Whenever the reducing equivalents are produced, what is the enzyme comes to the picture? That is dehydrogenase enzyme. Got it? Where the substrate level phosphorylation occur? Nothing but in the succinyl coenzyme A. When it is turning into the succinic acid, it is forming the substrate level phosphorylation. Got it? The only carbon compound, 5 carbon compound is the ketogluteric acid. So just remember this. Tomorrow we will discuss in a elaborate way, in a very clear. I will write the cycle for you, students. So if you have any doubts, please let me know in the comment section. And tomorrow we will discuss how many ATPs are produced. Likewise, we discuss in the glycolysis now. Like that, we will discuss about the Krebs cycle also. Then you came to know about for one glucose molecule, 38 molecules of ATP are producing now. So how they are producing, we'll see in the tomorrow's class. Got it? And we'll discuss about the steps of the Krebs cycle also. And why it is called the Krebs cycle? On the memory of the scientist name called Krebs, Hans Krebs, we are calling this cycle as a Krebs cycle. Okay? And it is also called as tricarboxylic acid cycle because on combining the acetyl-CoA, oxaloacetic acid becoming the 6 carbon compound, that is citric acid. In the citric acid, tricarboxylic acids are present, that is why it is also called as TCA cycle. Yeah? Okay? Then it is also called as citric acid cycle because the first stable compound is produced in the Krebs cycle is the citric acid, right? That is why it is also called as citric acid cycle. It is also called as Krebs cycle, TCA cycle and then citric acid cycle. Okay, got it? So this is about the today's session. Students, let's meet the tomorrow session and then I will elaborate you, tell you about how the glucose is producing the energy in the Krebs cycle. Where the Krebs cycle is happening? In the mitochondrial matrix. Why it is only happening in the mitochondrial matrix? Because the end product of the pyruvate, is getting converted only in the mitochondria because the enzyme name called pyruvate dehydrogenase enzyme present in the mitochondrial matrix. Then that 3 carbon compound pyruvate is converting into the 2 carbon compound acetyl-CoA. Then it is combining with the 4 carbon compound of acetyl-CoA, oxaloacetic acid. It is forming the stable compound called 6 carbon compound called citric acid. Right? That is why the first stable compound we call it as citric acid cycle. Got it? Please revise this and I said the mnemonic also do revise that. I will tell you the next tomorrow class how it is the forming one glucose molecule, how it is forming the 38 ATP molecules. We'll see in the next class. So before I am winding up the class, students, I would like to tell you very important news for you here. So here we are from NEET World, we are offering the long term courses for just affordable prices. The only limited seats are available, so book as early as possible because it's just affordable price like that, for the tuition fees is like that way. So it is just we are offering 3,333 for if you are studying the 11th or class 12th. It is the best opportunity for you because some people not read in the 11th and 12th, after they will take the long term. It's not the smart choice. Why? Because along with the IP, if you cover this coaching, then it is very the good attempt for your upcoming NEET and EAP set examinations. And for the droppers, if you are, you don't, for the droppers and repeaters, it is also best opportunity. We are just giving for 3,333. For class 11th and 12th and droppers, it is the best power step to, to take the coaching here, because we are offering just the price of 3,333. If you are really passionate to determine to see yourself as only doctor, this is the best platform for you. So what are we waiting for? Join to us. If it is both offline also and online also. For offline people, you can happily come to our institute name called NEET World, present in Narayani, Hyderabad. If you want to join through online, we have an app name called MedWell. There we will give you the access. So how you will get access? By calling this number. By calling this number, we will clarify all your doubts, and then we will give you access in our app name called MedWell. Daily DPPs will be there, periodic assessments will be there. So for the competitive examination, you have to write many mock tests. Then it will be, you will get the boost of, you will get the confidence, and you will clear out your examinations. As we are providing for the basic to the higher level, so it is the best platform. If you have any doubts, see our NEET World channel. There will be a plenty of demo classes are there. See that and contact us. Then it is a, then it will get clear picture come to you with you. So this is the best choice for you. And so because we are offering at the price of only 3,333. Okay, this is about our side information, and also don't forget to join our Telegram group also. As I said in the first slide, at the first only I said to you, by scanning that QR code you can access our Telegram group channel. So all faculty is there only. If you have any doubt, please post your doubt on the chat option. Then we'll clear you out all your doubts. So let's meet in the tomorrow session. Thank you.